Hold on.
Oh.
I gotta practice. Old genome pre-programming. Oh, sorry.
That's okay.
Kick the genome. Kick the genome. Kick the genome.
Congratulations!
Kick the genome.
We were stuck in this one for 45 minutes.
I heard that.
45
Are you sure you want to take this one?
Yeah.
Let's get over it someday.
We're about to make a transgenic plant. I mean, simply. It's not so simple. We're actually using transgenesis through some agrobacteria that's infecting the flowers of the plants we're dipping into the agrobacterium, infecting them with something called zinc fingers. The zinc fingers don't change one thing. They change hundreds or thousands of different genes by up-regulating or down-regulating genes that even may not be active in the genome right now. They may be hidden in there in deep time, like waiting for a panicked event or some kind of buffer to go down, like, oh, too much stress, and then, whoa, hairy, or like elbowy or bloopy, or whatever. And the down regulators also can turn off things that haven't been turned off in so long that it makes other things come on going, what the hell, what happened to our... Anyway, so we're going to up and down regulate genes in the thousands in these plants, in these seeds that are developing in the flowers. Up and down regulate them at the same time. They're competing for the same spots, thousands of spots. Will we be down regulated? Will we be up regulated? Well, what happens? The plant's genome goes like this. Up and down. All at the all in this one sort of 9 base pair area. I'm not sure which area we chose, which base pairs, we'll find out. Right now, we're about to infect some Arabidopsis flowers. They're like mustard plants. They're weeds. They're native. We're going to infect them by dipping them in a liquid that's filled with agrobacterium. And in the agrobacterium is a vector, and the vector inserts the zinc fingers into the plant genome. It goes into the flowers and into the gonads of the flowers, the balls of the flowers, the ovaries, and makes those new seeds and those embryos that are growing in the flowers altered. The way that zinc fingers alter the plants is not genetic modification, but actually they severely finger the genome. They actually push the genome really hard. They kick it, right? They kick it sometimes in thousands of places. We're adding 2 zinc fingers, 2 modules, 2 9 base pair modules. Do we know the 9 base pairs? These 9 base pairs, anywhere they exist in the genome, there's going to be a competition for whether they're upregulated or downregulated, which means expressing more or expressing less in all these areas in the genome. So we're gonna like kick it in 2 directions at once.
If you take that particular zinc finger we're going to dip today, and you fuse VP16 to it, which is the activation domain, it looks like this.
That's pretty different than your standard Arabidopsis.
Yeah.
Has it got way the freaking bigger leaves?
Well, yeah.
Yeah.
I mean, look.
What else is different? Because you have a better eye for Arabidopsis than I do. What do you see here that's unusual for this plant, phenotypically?
It's huge. I mean, and the leaves are huge. The rosette is huge. I mean, it's gigantic. If you look at this, this is more or less the Arabidopsis wild type, and you can see the difference.
Holy shit!
They're in a similar stage. Okay. It's called 16VD.
But what's VD stand for?
I have no idea.
For me, it's venereal disease 16.
Oh, sure, venereal disease.
But it makes for growth.
So just to give you an impression.
The cojones on that VD16. This is the standard Arabidopsis thaliana, workhorse of molecular biology when it comes to plants. Before rice, before wheat, this is the first plant that had its genome sequenced, right? So it's like the initial transcript of plant. For some reason, I think there's some kind of Christian thing about the mustard seed, But these are mustard plants. They're brassica, right?
Brassica.
If you want to taste
Exactly.
one,
Sure.
I don't think this is the place to taste it. Have you tried it?
No, I haven't. No.
Really? Would you try this one? Or?
No, no.
I have tasted them, and they're yummy.
We have to remove the flowers, and we have to make sure we don't remove the flower buds.
Right.
Because those are still suitable for dipping.
And what does that mean? They do open after the bacteria is on them, and then they probably fertilize after the infection.
Exactly.
So
yeah.
does the infection infect the eggs, like the ovaries of the plants?
Yeah, I think they infect embryos.
So once the fertilization happens, the bacteria is already there and it's ready.
Yeah. That's it.
I'm plucking any flowers that are already open. Yeah, so we're taking off all the grown flowers because Arabidopsis often, see what I just did was horrible. Look at
Fucked up.
I pulled off 3 buds with 1 flower. This is the flower. These are the buds that we're trying to not pull off.
You want to use it.
It's okay. This is sort of fun for me. Strangely fun. I've done a lot of farming, so
Maybe we should help.
These two.
zinc fingers sometimes just knock the hell out of the genome. Just actually whack it like with a stick. Like, like hit it in every direction, and what you have is that the buffer of the organism, the way that it works, is so confused that it actually has to find other pathways to work with. So the phenotype doesn't always change just because you change genes. But if you
yeah.
smack the hell out of the plant or stress its genome,
Yeah.
you get unusual expression patterns.
All organisms have some kind of plasticity, I think. So I think they're also plants are able to somehow deal with these disturbances.
And still retain
Yeah.
their original phenotype.
And I mean, it's a dynamic environment, so I think,
It takes a lot to make a new phenotype. But these zinc fingers seem to be slapping them into new phenotypes, sort of, right?
yeah.
That's cool. It's like, go ahead. You're free. It's sort of pre-designed. The punishment is designed in the architecture, so you're free to do whatever you want. Plenty. So we have these buds, and the bacteria gets on the buds, and then whatever's in the bacteria gets inside the embryos when a lot of these self-fertilize. So as they're open, they form pollen that fertilizes themselves, and the bacteria is right there to slam its load into them, to just be like, take this, and it's got like a little disease in there, a little genetic modification that helps it insert into the genome, but it's not inserting just genes, but instead it's inserting these zinc fingers, which are actually proteins. This is sort of a weird way to get around the legal problem of genetic modification. We're using gene transfer technology to get these proteins into the genome in this case, but there are ways to get zinc fingers into the genome and alter the hell out of the genome without doing any genetic modification. Is it protein
therapy.
Protein therapy.
But this is not protein therapy. This is genetic modification.
Because we're using a vector to get the protein modification in.
The genes for the.
The genes that we insert encode those particular proteins.
DNA is deoxyribonucleic acid, so it's an acid therapy. And protein is just protein. Because protein can work to alter the genome, but the protein itself is not genetic modification. It's proteinic modification or proteinic therapy. It's a fine point, but legally it's not GM. These are GM, but legally if you don't use vectors to get the proteins into the organism, you are clean, or legally, at least legally not even covered yet. So I don't know, how does it work? It's not suspect yet.
The ultimate goal would be to, for instance, take a solution of the proteins, the effector proteins, and spray them onto a plant, for instance, and then maybe those proteins go inside and do their job without having to insert a gene into the plant encoding such a protein. That's basically
a procedure.
yeah, and it doesn't involve inserting a gene into the plant, so that means it's not GMO.
Okay, once we dip these plants, how long do they grow until they have seeds?
I think that'll take a couple of weeks, maybe 2 or 3 weeks.
And then if we should see some upregulation and some downregulation. So some difference even from those giant plants you showed us, right?
Yeah.
Interesting. These are the flowers in the garbage. The flowers of death, the sadness. Yes. Those aren't the flowers of evil. Those are the wild type. This zinc finger is going to land with an upregulator and where it's going to land with a downregulator. Yep. And it's kind of hard to figure out where they land, like which lands where, right?
Yeah.
Is it impossible to figure out where?
No, it's not impossible.
But it's something that makes it at least harder for you to figure out.
Yeah.
We're actually doing something that hasn't been done before.
Yeah.
But in the name of art. You all right with that?
Yeah, I am.
I mean, are you going to try to get data from this too?
No, I'm not.
You're not.
I think pretty.
You're letting this
I know what's
project
gonna happen.
go.
Yeah. No, I'm not letting it go. I mean, but I'm not taking notes and stuff.
Yeah, it's the same marker, so there's no way to tell what's landed where. No, it's all right. You know why?
You still can't tell where it landed.
You can't tell.
No,
where
you
it
can't
landed.
tell. No, you just can tell it landed.
That's somewhere in the plant
yeah.
there is this or there is that. So we won't know where.
No.
So there is some jazz. Should I not lick it?
Don't lick it.
Why not?
Well, you can. I think
professional.
you could lick it. I think. I mean, it has been shown it can transform human cells.
Yeah. So if
HeLa cells, not human at all anymore. I think.
Right, but it could actually get into my. Like if I rubbed it on my balls, it might get in there.
I mean, the promoter should not be active in humans. So.
But if I put my sperm in there and let it grow for a couple of days and then put it in someone else, I might have a really big leafed girl. This is a detergent, and without this, the agrobacterium wouldn't be able to get in because it disrupts the waxiness of the leaves. But it's like basically what? Like laundry soap.
Pretty much, yeah.
Yeah.
Detergent. Yeah. It's a sort of like a soap, yeah.
Here's to Sylvius 77.
Oh, we were inside.
Can I glove up and get the 50 mils? What's that? Oh yeah. Where are they at? It's not so hard, hey? 50 or 15?
50.
I got it.
5 and 0. 50 Cent is starring in a movie.
Really?
It's coming up.
I don't want to, so that the competition is stiff between up and down regulation. This is beautiful, actually. You don't usually get so much product in molecular biology. You know what I mean? Like, that's a lot of product. The Falcon tubes.
Over here.
Should we label them? Well, yeah, VP16 is the one that overexpresses. And EAR is the one that represses or underexpresses. This would be manic. And this would be depressive. How many E's in depressive? So this would be... Just like depressives get more done before they kill themselves than manic people get done ever. The manic depressive dance. Manic depressive. Yeah, we'll put in some rap with this. Agitating.
Maybe California Love or something.
Check the clones.
Yeah, look fine.
I see some.
Oh, this looks fine.
All right, good enough. It's milky. It's thick and milky.
Back to the plant.
Back to the plants to dip. We're gonna dip the buds in this.
We're gonna take.
And the detergent's gonna irritate the wax membrane, make a sort of cut, like I have in my hand. Geez, I hope there's no interaction.
I think it's these nuts.
These nuts?
Yeah, that's right.
Watch your back, sir.
That's it. These nuts. Listen, I think I can never really understand what they're doing.
Yeah. The chronic man.
To a plant.
Yes. There he is. Yeah. Shall we glove up? Slide into things. So this is the place where it goes down. We're going to be infecting these little tiny buds. It seems innocuous, but it's actually inoculation.
So I think our final dipping volume should be like 250 mLs.
Mm-hmm.
So that means that we have to dilute this a little bit.
I'm calling it a bipolar flower because this is a manic and this is a depressive construct that we're entering into the buds. The zinc finger is 9 base pairs long. The same 9 base pairs and the same 9 base pairs here and here. Those 9 base pairs, anywhere they happen to exist in the genome of these plants, these zinc fingers can and may land. They're fighting for places to land. Anywhere they happen to appear in the genome, it could be hundreds of places, even thousands of places. It lands and it says, express more, turn on or turn off, whatever comes after any of these thousands of places. Just do it. They call it whole genome reprogramming. We're promoting and repressing thousands of downstream events at the same time. Here we go.
We dip the suckers. We dip those mofos.
See all that dripping? Nice. Actually, it bites them a little bit, like it hurts them. It causes a scar, like it causes a crack in the flower buds. And it's kind of like a wound or like a scratch. Kind of just.
Because in nature, the agrobacteria is actually not attacking this plant. It's attacking a lot of other plants, but on this one. So we have to help them a little with the detergent.
We're actually doing a transgene infection. We're causing mutagenesis. Mutagenesis on like a genome-wide scale. Inside the sort of not even fertilized embryos of this plant. We're starting a process that will probably overnight or over the next couple of days will fully realize itself inside the future generations of these plants. So these plants may be fairly normal, maybe, not showing anything, but the seeds of them and the seeds of those seeds and the seeds of those seeds may carry a whole genome reprogram, or what they call a whole genome mutagenesis, that involves thousands and thousands of different alterations of gene expression. Well, it could be lethal. Is that a revolt? Like, fuck this, I don't want to be this. This may be a revolt. When we talk to Bert, it's kind of interesting. This may be what pushes this plant beyond its normal expression patterns and into emergency reserve genomics. Like the buffer's gone. I don't have anything else to do but freak out, so I'm going to show some genes I haven't shown in a million years. It is very lovely. Like, this is almost real poetry. God damn it.
Transformance takes at least a month or two, I guess. The flowers have to open, seeds have to be produced, then the seeds have to be dried, and then you can plate them on selection medium. And then finally you can take the primary transformants, transfer them to soil, and then you have your plants.
My funny Valentine, sweet comic Valentine, you make me smile with my heart. It's easier to hurt than it is to help, right? You have to mush those organisms, put them on the edge. I don't know what kind of plant we're getting, right? I don't think anybody knows. We haven't done this before, so it hasn't been done before. We haven't mixed these kind of zinc finger constructs up and down regulated in Arabidopsis on this exact 9-digit zinc finger before, and so we're going beyond science, but not in the name of science. We're just doing it to mush the fuck out of the plant's difference and assess the difference on an aesthetic level. Like, what's up with you now? You know, like not like what's really happening. We want knowledge from this or utopian efficiency and progress. We just want to know, how's it going after that total kick and push smackdown? How are you doing now? Look at Nature Magazine this month. Biomedical supermodel. How do I do this? There you go. Look at it. It says there's been germline transmission in a transgenic non-human primate. They know how to genetically engineer or intentionally genetically modify the entire inherited cascade of this monkey. It's a biomedical supermodel. And this is Nature, what? May 2009. Big shit, that's been 3 years. What the fuck are we doing?
Ready? Wow. Fantastic. Let's open it. Oh, and open it again.
That was super finger